Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Welcome back to Neuroverse. Today's episode is another addition to our series, Tools for Looking into the Brain, which is supported by Scientifica. And today we are joined by two early career researchers to talk about their journeys into research so far and the importance of community in launching independent research careers. We're joined by Ashlan Reid, who is a postdoctoral fellow in the Zador lab at Cold Spring Harbor in New York, who is studying sensory processing in the cortex, as well as Mari Sosa, who is a postdoctoral fellow in Lisa Giacomo's lab at Stanford University and studying learning and memory in the hippocampus. Welcome to the podcast, Ashlan and Mari.
Thank you to both of you for hosting us on the podcast.
To get started, can you tell us a little bit about how you both got into the field of neuroscience in the first place?
Yeah, well, my journey into neuroscience, I think, started during my undergraduate at Harvard. I originally thought I was interested in molecular biology or maybe medicine, but as freshmen, we had an opportunity to take a small seminar course of about 12 people. And one of the ones that was listed was called Neurophysiology of Visual Perception with someone called Dr. David Hubel. And I had no idea who that was at the time. And I had no idea what neurophysiology of visual perception meant. But as I was reading the course description, I was like, wow, is this saying that, everything I see and perceive in the world is the function of neurons in my eye and in my brain, and that, the way I perceive the world might not be exactly the same as how someone else does because it's all just biology? Like, that's wild. And it was kind of a lottery system for the course, and I actually didn't get in. But then I saw that there was an opening posted on the website. And so I just emailed the professor and I said, I'd really love to take this course. Can I take it? And he was like, oh, yeah, sure. Which I think we're talking about like career advice, like always advocate for yourself. You lose absolutely nothing by sending that email. And I'm super glad I did. And it was kind of amazing. So obviously I then realized that David Hubel was a Nobel laureate who had won the Nobel Prize for discovering how cells in the retina and the brain process visual stimuli and transform that into visual perception. And he spent hours and hours with us just teaching us basic neuroscience, showing us his, original recordings from anesthetized cats where they discovered that as they changed the slide on the projector, that was the stimulus that would finally make the neuron fire when they had, everything else they had tried had failed. And so this is how they discovered orientation tuning, or how neurons respond to different orientations of lines. And then I just thought that was like the coolest thing I'd ever seen. And that I decided that this is what I wanted to do. I wanted to figure out how the brain worked. Yeah, I think that's where it all started.
What a person to learn that from. That's crazy, because I had a similar experience of finding that very interesting in my undergrad, but I did not get taught by Hubel himself.
No, I was incredibly fortunate, and even fortunate that I ended up at Harvard at all. They have an amazing financial aid program, which I hope is still the case, but it really made a difference for me.
And a really good demonstration of how early experiences really shape neuroscientists' futures. How about you, Ashlan?
So I also had my first exposure to the field of neuroscience as an undergraduate. But I was a biomedical engineering major at Duke University, and I had been selected to be on the medical devices track. So I was taking all of these electrical engineering classes. And then I decided to take a neuroscience class. And all of a sudden I learned that biology had found a way to build little electrical components and circuits to sense the world and interpret it. I learned that cell membranes act like capacitors and that protein channels in the membrane act like resistors. And ion pumps create charge differences across the membrane that function like a battery. And all of this circuit analysis and math that I was learning to do in electrical engineering could be used to model how neurons actually function in the brain. So things like Kirchhoff's laws, differential equations, this complicated math could be used to predict how neurons integrate synaptic activity, how they function as low-pass filters, how these special proteins making up the ion channels can actually sense the membrane voltage and perform operations like input inverting. And then all of these tiny little biological electrical components talk to each other, signal each other, work together, communicate, and allow the brain to build a dynamic internal representation of the outside world. And I just thought that was mind-bogglingly interesting, and something I wanted to know more about and spend my life researching.
Amazing, so cool. I always enjoy hearing an engineer's perspective or a physicist or computational neuroscientist perspective on the biology of neuroscience. And it's with extremely good timing as well, because we recently hosted this AI and machine learning versus brain function panel event. And we made a lot of comparisons between the circuitry of an electric board or the code that goes into AI and as well as the brain. And I recently learned about memristors, which blew my mind because they kind of act like LTD and LTP in the brain. So that was an absolute game changer for me. So I always find it cool when I hear different fields' perspectives onto neuroscience.
Yeah, I think the definition of computational neuroscience is just exploding in all different kinds of directions. There's the biophysically realistic models. You choose kind of a level of abstraction when you try to represent, how the brain functions with mathematical equations, with like concepts that we know from engineering. And I just think there's a lot to be gained by bringing in diverse perspectives like that.
Absolutely.
Could you tell us a bit about how you then transitioned from this original attraction towards neuroscience into your respective research topics?
Yeah, so motivated by, I don't know, this fascination with spiking that I had gained during that freshman seminar, I wanted to gain research experience. And there was a lab down the hall that studied visual processing in macaques and monkeys. And so they essentially have these classic kind of moving dot paradigms where the monkey has to perceive the motion of the dots and make decisions about which way the dots are moving. And I emailed the professor, Rick Bourne, and joined the lab to start doing some undergrad research. But undergrads don't typically work with monkeys. It's kind of too high stakes. And during summers prior to that, I had gained some research experience doing molecular biology, which I learned through that I really enjoyed being in a lab, but I found molecular biology itself a little bit unsatisfactory just because I couldn't really see what was happening. But nevertheless, my undergrad PI wanted to kind of leverage that molecular experience to let me ask whether there were molecular markers of visual feedback neurons in the mouse brain. So these neurons that project from higher order visual areas to lower. And this process required that I had to learn a bunch of techniques on my own. I had to order equipment and reagents and set up kind of a whole branch of the lab working with a senior research specialist that they had, which I think was more of a graduate student-like experience than most undergrads get, which was, I think in retrospect, really cool because I got to experience the early stage troubleshooting of a new research project and deal with failures and form my own hypotheses. But throughout this whole process, what I found the most interesting, honestly, was like watching the grad students and postdocs record from the monkeys because you could see spikes on the screen. You can plug the recording system into a speaker and you can hear them. And I really wanted to do that. So when I was applying to grad schools, I interviewed with someone named Lauren Frank at UCSF, and his lab studies rats, and they do electrophysiology with rats. So they're recording spiking while the rats are learning and making decisions. And he told me about this phenomenon called hippocampal replay. So this is where a group of neurons will spike together in close succession in a very compressed time frame, reactivating the same neural sequence of firing that the animal experienced earlier in the behavior. And so this is actually a mechanism of memory retrieval and consolidation. Like this is exactly what you would want if you want to study how memories are formed and recalled. And I thought that was incredible. So I joined his lab and I studied replay and how neurons in the hippocampus, when they replay, activate neurons in downstream brain structures like the nucleus accumbens, which is a brain area that's important for reward learning and encoding action value and helping the animal make decisions. And all of that, I thought electrophysiology is a really powerful tool for studying communication across brain regions like that. But one of the things that I was lacking was the ability to see the neurons that I was actually recording from, know which cells I was recording from, which cells they connected to, how they change over time. And so when I joined my postdoc lab, I wanted to learn a technique called two-photon imaging, which I know you've had some excellent episodes on the podcast before going into detail about that I'll refer listeners to. And this technique allows you to see the neurons you're recording from. And so I do two-photon imaging now in hippocampus, where I can track a whole population of neurons over a very extended period of learning for the animal, see how they integrate new information as the animal learns. We can also combine the imaging of their calcium activity with other fluorescent sensors for either things like neuromodulators or readouts of plasticities like cyclic AMP, or we can label the neurons based on their genetic profiles or where they project and really see in real time how neurons in the brain are integrating information and communicating with each other. And so I still study that same general question of how the brain forms and retrieves memories, and I've taken a specific focus into reward, because I think it's fascinating that if you think about any of your strongest memories in life, they're always your most positive or your most negative. And I'm really interested in that positive aspect. Like, how does a rewarding experience become amplified in your memory above other neutral experiences? And how do you layer information about new rewards onto stable representations of the external world? So let's say you encounter a new coffee shop that you've never been to before, and it has this very rewarding aspect to it that's similar to all the other coffee shops that you've experienced in your life. But now you have to add this layer of information about knowing that that's your new favorite coffee shop on top of your map of your neighborhood, and keep those things kind of parallel but separate. And so that's kind of what I study in my research now, and is something I'm interested in continuing in the future.
That's so fascinating. And I'm always impressed when people say they're doing two-photon imaging in the hippocampus because it's such a deep structure compared to the cortex where it's more commonly done. Yeah, what a great tool and a really interesting use of two-photon imaging and linking that all the way back to behavior. As far as I'm aware, Ashlan, you do something along similar lines, but using slightly different techniques. And maybe you could expand about how your first experiences with neuroscience evolved into a whole career.
Yes, I have actually several parallels with Mari's experiences, actually. I also joined a neuroscience lab as an undergraduate. I think that's a really important type of experience to seek out and to look for a lab culture that kind of matches and mentors young budding scientists. And my lab was also actually doing incredibly careful and smart electrophysiology in behaving macaque monkeys. But as an undergraduate, I did help build some of the setups to control the behavioral trials like Mari was talking about. I had little devices that would flash the lights or like read out what the joystick was doing when the animal made movements. But to do actual experimentation in my own hands, I was assigned to work with rodents, similar to Mari, and with an amazing graduate student. And like Mari, we were doing electrophysiology. So our project was to detect and treat seizures in real time. We were recording local field potentials in the cortex with depth electrodes. And I built an entirely analog closed loop device to detect the start of a seizure and trigger stimulation, like basically a small electrical shock, not painful, just kind of a tickle to a cranial nerve so that the seizure would stop. And it turns out that's a super effective technique to making seizures stop. And because the shock was only delivered in response to the seizure starting, the total amount of electrical stimulation to the nerve could be reduced. But also we were doing these electrical field potentials and recording seizure activity, which is kind of a conglomerate thing. So like Mari, I was also interested in moving beyond kind of this everybody talking at once, this local field potentials, which are great in time resolution, but are really limited in spatial resolution. You can't see the components, you can't see the individual neurons contributing to this kind of global brain activity. So I wanted to see how individual neurons and neural networks were functioning dynamically in the cortex, how that activity was distributed in the cortical layers. So I chose my graduate and postdoctoral work to be in labs where both single cell electrophysiology and network dynamics were the focus. So in my postdoctoral work, my favorite set of experiments that I've done recently, I imaged voltage sensing dyes across the layers of the cortex, in the auditory cortex, to watch where axons from the thalamus make their first synapses as they arrive to deliver information to the cortical layers. These dyes, voltage sensing dyes, get embedded into cellular membranes and change color depending on the voltage across the membrane. So in contrast to what Mari was looking at, calcium imaging, where you need spikes or even bursts of spikes to get a signal, voltage imaging actually allows you to see subthreshold activity in space and time. So you can really detect where the axons are making the voltage change first in the postsynaptic membrane, where those synapses are actually located in space. And importantly, the brain develops. As the brain develops, axons from the thalamus need to migrate up to their final destinations in the cortex. So they migrate in space, and they need to migrate properly to set up the sensory maps in the cortex to process, for instance, sounds in the environment or some other sensory parameter that's going to be, tone frequency or the direction that the mouse's whisker is deflected or the visual stimulus in space, like the classical visual receptive field experiments. So these axons need to migrate properly to set up the sensory maps. And I'm interested in what kind of electrical and molecular signals help this to happen properly. How do axons get to where they need to be? And what goes wrong in injury and in neurodevelopmental disorders? So like you were saying, early experiences are important. It's important that the animal's experiences when they're young and developing play a big role in how the adult brain functions. And to circle back to my own formative experiences, when I was young and getting started in neuroscience, I've started to study seizures again because I'm interested in how juvenile seizures impact how the cortical architecture gets built. So in these classes, we're taught that neurons that fire together wire together, but seizures are like the ultimate fire together situation. So I'm studying a particular mouse line that has seizures all the time when it's going through its developmental period. But this particular mouse line is a great model for human absence epilepsy, but its brain still looks pretty normal. These mice sense things, they behave, they eat, they make babies and raise them. Why aren't their brains just an overly connected mess? How can the rules that are connecting the neurons tell the difference between synchronous activity being caused by stimulating the environment and synchronous activity being caused by a seizure event? So I'm really interested in discovering the homeostatic mechanisms in place to govern the connectivity rules while still allowing developmental mechanisms to build a brain that functions. And so understanding the links between electrical and molecular signaling programs is a big part of my future plans.
Wow, so cool.
That was actually such a moment for me because you basically described also what I study. So it really spoke to me because I'm looking at activity-dependent plasticity and these homeostatic mechanisms during development as well. So I definitely share that passion, and I like how you came all the way from an engineering background and kind of through all the different techniques landing on voltage imaging, which is so cool.
But kind of old school. I mean, it's a little old school, but it really is the best technique for my purposes to answer the question. And so I feel like that's a big part of our message today, which is your technique doesn't have to be new and flashy. Sometimes it can be something that's kind of an accepted, strong, basically hypothesis-driven question, and you find the technique that will help you answer the question that you're looking to answer. And so sometimes it can be a new flashy technique, and I love building new things, but sometimes it can also be kind of the old school voltage sensitive dyes where you see, they're faster than calcium imaging. There's a trade-off, right? Calcium imaging is super powerful and the signal to noise ratio is way better, that there's just, trade-offs. And subthreshold imaging where you don't require spikes, it's a reason. It's a reason that you make one choice experimentally over another.
Yeah, that's a really good takeaway to not just use techniques for the sake of using them, but in order to answer specific questions. I wouldn't see it as old school, but I don't know, I'm much more new to the field of neuroscience. I always see voltage imaging as this amazing kind of newish thing that is currently being really developed, and there's new dyes all the time, but maybe I'm wrong. I don't know.
Well, it's getting those things to be genetically encoded. The dyes where you stain the tissue, you can do that in vivo, but that's largely an in vitro type practice where you make a living brain slice, an acute brain slice. And you can actually do that with thalamocortical axons. You can make an angle through the brain that preserves the axonal connectivity. So both the neurons and axons in the thalamus are living in the preparation, but also neurons in the cortex that are receiving these synaptic inputs are living in the preparation.
Wait, sorry, I just wanted to clarify. So as in making the voltage sensitive dyes genetically encoded is kind of the current challenge and is what is being developed now?
Voltage sensors have some pretty substantial challenges. And I think the field is really fastly converging on voltage type biosensors that can be genetically encoded. But I think that's where you see this disconnect because of all of the cell type specificity that you can work into like a molecular approach, that kind of thing. So dyes go in everywhere. They just stain every cell membrane that they can. and they can actually have some pretty important effects on like capacitance of the membrane, that kind of thing. So I think that the field is moving towards new flashy invented sensors that are getting better and better. So I don't know that we're there yet, but having genetically encoded voltage sensors is on the horizon.
Yeah, I can add to that a little bit because we've got, some of the leading research on genetically encoded voltage sensors at Stanford. For example, Michael Lin's lab, which the Giacomo lab has collaborated with in the past. And my understanding of the main limitations is both that you need a bright enough signal to noise ratio of individual cells. You need high enough sensitivity where you can see essentially the readout of individual spikes. And because you're recording voltage signals, which are much, much faster than calcium signals, you need a very high sampling rate. So with calcium imaging, your imaging sampling rate can be 15 hertz, let's say. It's like often what I use. And that's plenty to record calcium transients that have a decay of, 100, 200 milliseconds. But for voltage sensors, you really need to be recording more in like the hundreds to maybe thousands of hertz range. And that seriously limits your field of view size. So there's a trade-off in two-photon imaging between how many lines you can scan in a big rectangle that's your field of view and how fast the mirrors are moving to scan all those lines. And so, for example, the work that my colleague Mark Plitt collaborated with the Lin lab to test some of these early genetically encoded voltage sensors, he had to record essentially the width of a single neuron in order to get the scan to move fast enough. So there's this trade-off between you have the sensitivity to image individual neurons quickly enough with these voltage sensors, but you kind of lose one of the advantages of two-photon imaging with calcium imaging, which is that you can record many hundreds to thousands of neurons simultaneously. But I think there are recent developments that are going to kind of push those limits and overcome some of those challenges.
Yeah, I think wide field rather than multiphoton, but then you kind of trade off your resolution. So it's all speed and resolution. It's all trade-offs and pros and cons.
I find it interesting how Clara and I both have this perspective that voltage imaging is this brand new emerging field, and Ashlan, your experience with it is that it's actually an old school technique that's just getting like a little bit of a makeover and trying to come back again. And the things that you were saying about the trade-offs are really interesting. And I kept thinking in my mind, I know it's difficult, but like if subthreshold properties are so important for your specific experiments, why not maybe attempt in vivo patching? Because if sometimes the resolution is to the extent of a single neuron, you already get that with in vivo patching.
So that's exactly what I began my postdoctoral work doing.
Okay.
I went to Tony Zador's lab when the lab was still doing in vivo patching, and I have an entire manuscript worth of data that talks about patching neurons in auditory cortex and looking at the synaptic input. Because more than that, you can make some assumptions and simplifications and actually tease apart excitation and inhibition by doing some kind of mathematical tricks and voltage clamp. And so you're right that patching is very powerful and it lets you see inside this. It's really amazing. And it's why I wanted to join the lab, the main driving force, if you will. And it's just very low throughput. So when Mari's looking at thousands of neurons at a time and everything is kind of contributing to this overall perspective, it's really difficult to ask that same type of question to individual neurons and then basically amassing a population of neurons where the state of the animal may be changing or the state of the cortex might be changing or, any number of things that affect like translating a single cell's activity into this population dynamics, I think. So you, again, got to really figure out which approach is the best way to answer the questions that you want to get at.
Yeah, makes sense.
Yeah, like I guess very small structural scale for voltage imaging could be useful if your question was about subthreshold changes in activity, differences across a single neuron, like if you're looking at the subcellular level. So yeah, as you said, it's all about trade-offs. And I'm glad that discussion has kind of brought that to light because actually when I think about it, it has been a key theme across our Tools for Looking into the Brain series, where we're always discussing the pros and cons of each technique. And this is also quite a good segue to my next question, which is about challenges in your research journey. And I was wondering if you could share a little bit about what your greatest challenges have been whether that's technical or more personal, in your research journeys, and maybe some advice that could come out of that?
Sure. So I think a really big one for me was learning to dissociate experimental failure from personal failure. There's a lot of scientific failure in the scientific enterprise, and this is key to making progress because often the moments where we learn the most are when either our hypotheses were wrong, a certain neural activity pattern isn't correlated with what we thought it was correlated with, and these failures are great as long as you can learn something new each time. So as you're, let's say, troubleshooting a new technique, I've now come to learn that as long as I can always think of what the next step is to try, it's all good. And it was a pivotal moment for me in grad school when I finally realized that these failures didn't reflect that I personally had failed as a scientist, but that I was instead doing what a scientist needs to do. And we can't anticipate all possible variables, especially when trying to do something that literally no one has done before, which, for anyone thinking about going to graduate school, I want you to understand that the science you're doing is the real deal. Like you are the one making scientific progress, trying stuff that no one else has done, or maybe they have done it, but we're still learning how to do it and there's unanswered questions. And I remember in my first or second year of grad school, I expressed to my PhD advisor that I was so impressed with how much he knew and how worried I was that I was never going to know that much or be that smart. And he was like, Mari, I'm not any smarter than you. I've just been doing this for 25 years. Like, it's all just experience. Everything is just experience. And you're here to get experience. If I expected you to know everything already, then I should just give you your PhD. You'd be done, right? Like you're you're here to learn. And so I think that's a really important perspective to keep in mind, that the main thing you're learning in, let's say, graduate training or in scientific training in general is how to learn. And you become an expert at figuring out brand new things, at learning how to learn something that you've never thought about before. And so as long as you can kind of develop the confidence to do that, then it makes the failures a lot more weatherable because you know that you've been there before and it turned out okay. And there's always something new to try.
Yeah, I find that very relatable in terms of my experience learning patch clamp, because when working with technical techniques such as patch clamping, it's so much troubleshooting. And when I was first doing it in my master's, every single day I would come in, there's a new problem. And I remember thinking at some point, when are these problems going to end? Like every single one had a different solution. So when am I going to know all of the solutions? And to this day, I don't. I still can't find new problems when I'm using different setups. It's always an unexpected problem, but I now have a different attitude, as you said, Mari, that it's not about I'm failing, it's my fault, but it's more about, okay, how am I going to overcome this? It's like, oh, yet another challenge. I will not let anything get in my way anymore. Yeah.
Exactly, exactly. And even if you've done it hundreds of times and you thought you found the whole parameter space of all the things that could go wrong, like something new can always crop up. I think this is especially true with in vivo work where you're working with, living, behaving animals. Sometimes they just choose not to do the task that day, or sometimes, maybe they're not feeling well and you can't tell. There's just all these things that can come into it that you really don't have control over. And some of that is beautiful, like being able to study that individual variability in biology and, interact with the living system is one of the highlights. Yeah, so just I think being prepared for something new to always crop up, no matter how many times you've done it, but just yeah, gaining that confidence that like, okay, it's a new challenge, I'll overcome this one. And I think another piece of advice that I want to share is that if you can't think of the next thing to do, ask somebody. Like ask for help. There's so many people who have tons of experience that you can learn from. And while there's a lot to be gained, as we've kind of talked about, in figuring it out yourself, there are limits to that. And sometimes you can save yourself a lot of time by just asking, and there's never any shame in asking for help. So that's something I want to advocate for as well.
It's so refreshing to hear biologists, like people who came from a biology like training trajectory. It's so nice to hear you talk about biological variability because coming from an engineering perspective, I was just overwhelmed by the amount of variability and uncertainty inherent in biology because it feels like sometimes there's more exceptions than there are rules. And there's always going to be something that's impossible to control for or impossible to know that you're not controlling for. It always seems like there's more unknowns than there are equations, and that can be really frustrating. And sometimes, like Mari said, there will be experiments that just don't work. I mean, Clara, you said this too. There'll be experiments that just don't work and you don't know why. Or there's results that you get that are hard to bring into agreement with what has been shown before. And what defines success for me, I think we're all saying the same thing, is resilience and perseverance in the face of challenges like these. And just like you all have been saying, separating complications from personal failure is a skill, and it has to be learned, it has to be developed and supported and practiced. It doesn't usually come naturally to people, and to learn how to kind of strengthen that resolve. And I think a big component of that, to learning how to persevere, is to find support within the scientific community from people that you trust and who want you to succeed and who you can ask these questions to.
Yeah, both of what you, Ashlan and Mari, have been saying resonate with me so much, because I'm starting properly my PhD project. I finished my rotation year, so now I'm starting, and it's been 2 months of basically troubleshooting the behavioral setup. And let me tell you, it's not just biology that has uncertainty. Engineering has some interesting amount of uncertainty too. Hardware troubleshooting is one of the hardest things I've done. And so, yes, it is frustrating, and it's very reassuring to hear about your experiences and all of the amazing advice that you've been giving. And I need to start taking it myself a bit more, including the asking questions as opposed to just gnawing at the problem for hours on end. So, yeah, really good message.
I was lucky early on in my undergraduate experience that the lab and the surrounding labs had several postdocs and graduate students who taught me the importance of supporting each other and doing science together rather than in a vacuum. They taught me how to like navigate the tens of thousands of people at the Society for Neuroscience meeting, and they taught me how to present in small journal clubs. And I feel like they modeled resilience themselves. And they've all been just a tremendous influence in my scientific career and my personal life. So I watched them go on to launch their own labs, and I would happily hang out with any one of them for as long as they would let me. Because I think it's important to have role models that are as human as they are scientifically rigorous, so that you can see how people have kind of honed this skill of resilience.
Yeah, also a lot of people ask me, how do you choose your PhD lab following like a rotation year, or just in any case, how do you choose which lab you want to join for your PhD? And I think the community aspect of the lab is really important. And I am very lucky to be in a lab that has a lot of expertise floating around. And so it really helps to be able to feel like I can ask whoever about specific questions about different aspects of my project, because each person has, their area of expertise and is friendly enough to want to help me. And that has made a huge difference for my PhD experience so far. And not only your lab, but beyond the lab too, like in whatever center you're working in. If there's a sense of community and people are approachable, it really helps to gain confidence. I think like having conversations with other people in my center have also helped me, gain that ownership of my project, be able to talk to others about it and open up with the problems I'm facing instead of keeping everything inside and trying to hide all the problems. Because turns out that if you share that with other people, they often tell you that they are experiencing the exact same problems.
Yeah, definitely. For anyone deciding, what lab to join for their PhD project after rotating, I'd say the main two things are see how other people pick each other up. Like, how has the lab dealt with challenges? How does the PI deal with challenges for the people in their lab? Yeah, how supportive is that community, and would you have the resources that you need to succeed, even if everything is going wrong, as it sometimes does? And then also, like, is it a topic that you can get excited enough to work on for several years or maybe the rest of your career? And it doesn't have to necessarily be the topic that you thought you'd be excited about coming into, but it needs to be something very motivating, because getting through those challenges is much more doable if you can kind of keep your eye on the prize. Like, what is this big picture question that we're trying to answer here? And why do I think that's important? And can I see how what I'm doing could lead to that answer, even if I'm like deep in the weeds of the hardware debugging and the chasing down the random source of electrical noise that's contaminating your eFIS recording or whatever it is for.
Yeah, that's such a good point. And I think we'll wrap up this episode here, but stay tuned for our following episode where we're going to dive more with Ashlan and Mari about how to launch an independent research career and the importance of community for that. So stay tuned to hear that. If you enjoyed the episode as much as we did, and be sure to follow us on social media at neuroverse_pod and check out our website, neuroversepod.com. Thank you for listening.